especially creationists really like to quote this, but then they stopped the quoting here because, of course, this was kind of a poetic question from Darwin, and he already knew the answer, which is that yet reason tells me that if we can start with a very simple beginning, a humble beginning for the eyes, then his theory of gradual evolution by natural selection through variation, inheritance, and differential survival can explain even the evolution of very complex structures such as eyes or other sense organs or the brain. And the idea of Darwin was that maybe initially there was just really a very humble beginning, something that can be called Darwin's proto-eye, which was maybe just some sensitive area of an animal's skin, maybe with a little bit of pigment and an optic nerve, which was coated with pigment. And this could already give some animals some ability to sense light and react to light. And then through natural selection, we can then get to the evolution of more complex eyes. So this proto-eye is a very interesting question in evolutionary neuroscience. And if we think about the evolution of eyes, of course, we know how complicated our camera eyes are, or the eyes of, for example, arthropods, the flies. Compound eye is super high resolution eyes and very complex structures. But and obviously they have a great use for those animals, but where do they come from? So we can think about a conceptual series of improving resolution, where in each stage we just increase the resolution, let's say by twofold, and we start with some very rudimentary light sensing, maybe just an animal's ability to distinguish if there is light or no light, maybe distinguish between day and night, or a shadow or not shadow, or is there UV light in the environment or not. And then this could have evolved with some kind of shading pigment into a system where the animal could see, is there light on my left side or on my right side, and maybe turn to the light. And then we can imagine that by adding maybe more photoreceptors and more optical structures, so really gradual steps, we could increase the resolution subsequent series, and actually get even something which is as complicated as our eyes, or the eye of the eagle, which is one of the most highly resolved eyes in nature. So this is the theory of gradual evolution. And we know that evolution works. It's an algorithmic process. As soon as you have a population with variation and with reproduction and inheritance, evolution will automatically follow from that. It's like a mathematical rule. So there is nothing that needs to be proven. It's mathematical level proof that evolution works. And if there is selection in the right direction and sufficient variation, then we can evolve even complex structures like the eye. And the famous study from Nielsen and Pelger from 1994 made some theoretical calculations of how much time would it take to evolve from a very simple, let's say, light sensing sheet of cells through some invagination and the condensation of some kind of cells that would make a lens. How long would it take to evolve something which looks like a primitive camera eye? Let's say this would be something like the eye of a fish. And they just allowed 1% variation in the structure in every generation, and they could calculate that it only takes less than 300 steps, so 300 generations, if we have 1% variation, to get to a camera eye from just the light sensing structure. And we can imagine, as we are in these stages, every little bit of improvement in the optics can have a selective benefit. So natural selection can act on those variations and lead to an improvement of these eyes. The big question that remained is that what would an animal do with just the light sensing very simple structure, this Darwin's proto-eye, because obviously for natural selection to work, every single step has to have some kind of function and an improvement in the function. So, but you could say, well, an eye which just is a sheet of cells has zero selective function. Why would an animal need such a structure? So there's this big question of how do we start the evolution of an eye? Once we have started, then we now know, and there is very good theoretical and also comparative considerations, we can get to very high levels of complexity through gradual natural selection. So I am interested in addressing this question: what is the function and the usefulness for the animals of the very primitive light sensing structures that we still find in nature? And the good news is that actually Darwin's proto-eyes, or structures that are actually very similar to what Darwin conceptualized, still exist in nature. And they exist in large diversity across many, mostly marine organisms, and mostly the little ciliated larvae of marine invertebrates, such as annelids or flatworms, or the enteropneus, so these are hemichordates, or other animals, also cnidarians. So in a large number of animal phyla, we find these very simple eyes, and often they only consist of one or two or a handful of photoreceptor cells and a few pigment cells, as you can see in these schematics. So this would be a photoreceptor cell, and this cell with these black dots here, this is the pigment cell that provides some kind of shading and directional information to this proto-eye. So we are studying such proto-eyes in our favorite model organism, the Annelid Platineris dumerulii, and this is a marine worm that can be bred in the laboratory, and we have a big culture in Heidelberg, which is very far from the sea. And over the years, my lab and other labs could develop many tools, such as microinjection, transgenesis, neuronal promoters, and other genetic resources. We can do behavioral assays with large numbers of larvae, and we can do very high resolution neuroanatomy, as I will show. So what do the eyes look like in these little creatures? So first of all, I show you the spawning. This is the adult worm. They are about 3-4 centimeters, and they have this beautiful nuptial dance. The female releases the eggs, and the male fertilizes the eggs through external fertilization. So we can spawn them in the beaker, in the lab, in seawater. And then a few days later, we get these beautiful zooplankton larvae with little eyes that are swimming in our dishes, and we can do lots of experiments with them. Now, the early larval stage, this is a 2-day-old larva, has a band of cilia. It's an animal of approximately 150 microns. It has already a mini brain, and it has 2 eye spots. This is shown in red, the pigment of the eye spots. Here we are looking down on top of this animal. And this is called the Trochophora larva. It's a famous type of larva from zoology. So in the first study, when I became interested in eye evolution, I looked at how these eyes look like and what is their function. So this is a high-resolution reconstruction of this pigment cup of the eye. This is the pigment, and red, you see the light-sensitive area of this photoreceptor cell, the so-called rhabdom. This was in the schematic, these fingers where light reception occurs. And you can see that the pigment is shading the photoreceptor from one side. So this is a direction selective structure. It only sees light that comes from this incoming side, and the light from the other side is blocked. And this photoreceptor cell here is directly synapsing, so sends an axon to the ciliary band cells, to the ciliated cells, and forms synapses on these ciliated cells. So this is not like our eyes that project into the brain, then it goes to lots of interneurons and the visual center. This is a direct sensory motor eye. It's sensing light and also has a motor neuron function. It synapses on an effector organ, which is the ciliated cells. And what is the function of this? So the result of this arrangement is that the larvae can react to directional light with a behavior that is called positive phototaxis. So if we switch on directional light at the one side of a beaker or a cuvette, the larvae swim to the light in a directional manner. So they know where the light is coming from. And the way they are doing this is quite interesting. So they have to rotate in this helical fashion. So they move the body constantly in this helix. And as they do that, they are rotating their eye, which is shaded from one side. So they are essentially scanning the visual environment. And if there are differences in light intensity, for example, there is light coming from top in the ocean, as normally happens, then at every turn, there is a signal from this photoreceptor that goes to the cilia and slows down the beating of the cilia. And we could show through lots of experiments and computational modeling that this direct sensory motor innervation from eye to cilia is sufficient to drive phototaxis. And we could also show in this computer model that a spinning, which is this rotation of the body, is essential for this behavior to occur. So if we switch out the spinning, make it zero, then there is zero phototaxis. Whereas if the larva spins either to the left or to the right, there will be phototaxis. So very well understand now the mechanism. And this is the simplest eye that exists in nature, basically, that gives rise to a selective behavior. Obviously, positive phototaxis has a very important function in the life cycle of these larvae. It tells them where the sun is, so where upwards is in the ocean, so they can follow the light and swim up as soon as they hatch from the embryonic stage and develop these eyes. Now, one day later, they have developed a different type of eye, which are shown here in these black dots. And these eyes are a little bit more complicated. And they are, if you tether a larva and illuminate them, you can see that they lead to the contraction of the trunk, which means that if you illuminate here, they bend on the other side. And if you illuminate the other eye, then they would bend on this side. And this is also a type of phototaxis, but this is a slightly more complex neuronal architecture. We call this visual phototaxis. And here already the larva and the neuronal circuit is comparing the incoming light between the left and the right side. So in the early helical stage, the larva is just swimming, and there's a temporal comparison, right? As the larva rotates, the light level goes up and down, and that's how they react to it. But here, the two eyes actually compare the amount of light, and they will turn according to where there is more or less light on which side of the body. So we call this a 2-pixel eye. And I will show in a minute what is the neuronal substrate that allows this 2-pixel vision. So to get to that, we had to look at the neuronal circuit. So obviously nervous systems, neurons talk to each other through synapses and also through non-synaptic means. But in this case, the synaptic connections were very important. And so we acquired over the years this whole body serial electron microscopy data set of one such larva. And you can see here a reconstruction. And we were first interested in the neuronal connectome of the visual system. So how do the neurons connect to each other through synapses in this visual system? And so we had to reconstruct the larva. This shows here the nervous system. This is the 3-day-old larva. What you see in blue are the different cilia. So this is still a ciliated larva, but it already has muscles. And this is the typical rope ladder-like nervous system of the annelid larva, with the brain and the trunk, with the segmental trunk and segmental nerves, and this rope ladder-like ventral nerve cord. So we have the entire connectome of this animal, which is a connectome of more than 2,000 neurons. And connectomes, you may have heard from the Sinorhabditis elegans connectome, which was the first pioneering work done to map all the neuronal connections in the organisms. We have now connectomes for example, the Drosophila larva and the Drosophila adult brain and nerve cord and parts of, for example, the mouse cortex. And this is another connectome, which is a whole body connectome, where every dot here in this graph, network graph, represents a cell, a neuron or an effector cell, for example, a muscle or ciliated cell. And the connections represent synaptic connectivity. And these are arrows, so directional connections. You don't see the arrows, but if there's an arrow, this shows that this neuron synapses on that other cell with this many synapses. And we analyzed a lot of the subcircuits in the system. And what I'm going to show you now is only the circuit of the visual eyes here, which is, as you can see, just a small part of the entire nervous system. And this is dedicated to this visual phototactic function, this 2-pixel vision. So if we zoom on, this is again the behavior. Here is the structure, the photoreceptors. Now these photoreceptors project into the brain, just as you would expect from a normal eye, like a visual system, that there are projections into the brain, not like in the early stage, where in the helical phototaxis, where the eye directly projects to the periphery, to the effector. And there are interneurons, different types of interneurons and different layers of interneurons. And eventually the circuit reaches these motoneurons that are going down in the trunk and innervating longitudinal muscles that lead to the bending of the trunk. So here is the full circuit. And you can see that it's more complicated, but what is fundamental here, that between the left side and the right side of the body, there are reciprocal connections. And this happens at this interneural level here. These yellow blobs represent these yellow interneurons here that synapse on each other. So the left side synapses on the right side and the right side synapses on the left side. And we think this is the first fundamental connection to make this left-right comparison possible, to get basically this spatial vision where the animal can tell the left from the right input and then be able to turn through this motor circuit here, these red motor neurons that innervate the muscles here. So this is a complete visual circuit, and this was the first animal for which we had an entire visual circuit mapped, and we also know very well the function of this circuit. So now what we are very interested in, to explore further the diversity of these simple eyes across many different animals, and also look at other forms of light sensing, maybe even more simple than phototaxis, even subsystems without pigment. And so we are doing this in this large ERC funded project, where we will look at larger diversity of organisms, including Amphioxus and hemichordates and cnidarians and some flatworms. And I am just going to illustrate this with some examples and some of the results we obtained. So this is again just some beautiful scanning electron micrographs of the diversity of ciliated zooplankton. And we can find eyes in different orientation and configuration and in many of these various larvae. So the first project I will briefly introduce is the coral larvae. So corals also reproduced by these swimming ciliated larvae that look like this, are completely covered with cilia, but they don't have any eyes. And this is how they swim. They also swim in this helical manner. This is a little coral larva here. This is a part of its ciliated epithelium, full of cilia, and you can see the beating of the cilia here. They are propelling the beads in this experiment where the larva is tethered, and you see the beating of those thousands and thousands of cilia. And what was interesting to find that these larvae can also react to light. So in this movie, you see larvae in the dark. They're kind of sleeping, and then the light is switched on, And you can see that the larvae start to wake up and move and swim more and more. And so this is something which we call like a dark response, a scotophobic response. It's a phobia of darkness. So when there's darkness, they stop swimming. And what also happens when we switch off the lights, you can see in this video that the larva contracts. And so they become more, since this is light on and light off, they contract. And here's some quantifications. So this is the length width ratio. So basically to quantify the contractions, you can see that the larvae contract when the light is off, and then they relax and they contract again. And when you see this dramatic difference in the shape, and when this contraction happens, they also stop this very powerful beating of their cilia. We can see it here. This is the quantification of the flow fields around the larva. When light is off, the flow stops. And this is quantified here. So essentially, they kind of contract and stop swimming when in a shaded area. And we think that ecologically, this helps them somehow to avoid to settle under very dark conditions, because if they stop swimming, they will somehow float away from this place with unfavorable conditions. So this is also a very simple light response without any eye. It has photoreceptor cells, obviously, but they cannot sense the direction of light. They cannot see. They just react to light and darkness. And we are also trying to understand the neuronal mechanism of this, and we have an electron microscopy reconstruction done by Emily Broderick of one such larva. You can see here, it's a very simple nervous system. These blue cells form a nerve net, and we have some ciliated sensory cells in this larva. And there's really a very simple neuronal wiring here, which is underlying this light sensing and other behaviors. Another very interesting larva, this could be a little bit more advanced behavioral stage. This is also an Eiderian. These are the larvae of the hydrozoan Clava multicornis. And these larvae do not have pigments, do not have eyes, but they can still react to directional light stimulus. As you can see here, they swim towards the light with these wriggling motions. They are basically swinging their anterior pole left and right. And this is how they can distinguish, we believe, where the light is coming from through these head swinging behaviors. And probably through body shading, they are able to tell where the light is coming from. So this would be something like an intermediate stage of complexity, still no eyes, but through these head movements, the animal is able to tell the direction of light. Now, we also explore the diversity of larvae and eyes through field trips. This is a recent field trip from Ischia, where we collected some beautiful organisms, which we also will study in the lab. And this is what we have found. So we placed the slides, traps into the water a month before we went to collect them. And when we collected these microscope slides, we found the structures like this. It's approximately 2 or 3 centimeters. And what these are egg capsules. So each of these structures is an egg capsule with several eggs. And if we look into them, they are full of these amazing swimming ciliated larvae. And you see these pigment spots. So those are the eye spots. And when these larvae hatch, or we take them out from their capsule, then they are actually phototactic and swim to the light. And this is a scanning micrograph of one such larva. It's also fully ciliated, but this larva has eye spots, and this is the larva of a flatworm, a polyclad flatworm that lives in the Mediterranean called Planocera. So we are now working out the neuronal connectome and the mechanism of phototaxis in these larvae to have a perspective from another phylum, which is very different from the annelids. So these are the Platyhelminthes, the flatworms, and so how this look like. So we hope that we didn't make a mistake by bringing them back to the lab. So this is how these larvae look like. Very nice little larvae and very strongly phototactic. And we could collect thousands of them during this field trip. Now, what is already known about these larvae, that they have eye spots, and also the so-called brain ciliary photoreceptors, which I didn't have time to introduce, but we have them in Platineres, and we could show that these ciliary photoreceptors, they are also not pigmented, but they are responsible for UV sensing. They express a UV absorbing opsin, and they connect to another circuit than the visual system and can mediate downward UV avoidance behavior. And so this is also an interesting light sensing system which is not an eye, just brain photoreceptors, no pigment, no direction, no vision, but a very important behavior is mediated by these structures, which is UV avoidance. And these platform larvae, these Müller's larvae, also have both these pigmented eyes, these so-called rhabdomeric eyes with the pigment. You see here the pigment and the rhabdoms. And they also have these brain ciliary photoreceptors, very similar morphologically to the annelid ones, but we don't know yet the function of these structures in the Müller's larva, and this is what we would like to figure out. So one function is certainly phototaxis, but they may be other functions such as UV avoidance. And if we then compare functions and circuits across various marine phyla in these primitive eyes, then maybe we can reconstruct what was the ancestral condition and what, for example, the last common ancestor of all bilaterians had as an eye. And this is kind of our approach, this comparative evolutionary neurobiology and connectomics and functional analysis of visual systems to try to understand what are the commonalities, what are the unique features in different phyla, and try to reconstruct some pathways for eye evolution. So coming back to close my talk, coming back to this schematic, so we now know and have several examples of these very primitive or simple light sensing systems. And we don't claim that all eyes evolved from the eye of the annelid. Of course, these are all modern taxa, and they underwent as much evolution as us and eagles with the camera eyes. They just never bothered to actually evolve these complex visual systems, except from some outliers. Also within the annelids, there are some which are pelagic predators, and they have very complex high-resolution camera eyes. So it's not that these animals cannot evolve that, they just don't care, they don't need to, because they only care about light direction, phototaxis, or UV light, and these simple systems are perfectly capable of mediating those senses and behaviors. And depending then on the animal's niche and ecological requirements, animals evolved eyes of different degrees of complexity. Of course, an eagle's eye or a fly's eye is very expensive to maintain. So as soon as you don't need that eye, and this is observed, for example, in cave animals, you very rapidly go back and lose the eye. So actually, to maintain such complex eyes needs constant maintaining selection or stabilizing selection through evolution. Otherwise, the structures which are super expensive would immediately degrade back to simpler forms or would be lost. So now we could close some of the gaps in this evolution of eyes from Darwin's proto-eyes to the human eye scheme. And we now know that there are ecologically relevant and selectable, simple, very simple structures across many animals, and presumably also in the first vertebrates that started to evolve eyes. So eye evolution began with humble beginnings, and then through Darwinian gradualism and natural selection led to the complex eyes that we see today. So thank you very much for your attention, and I would like to acknowledge my group and our funders, and I am happy to answer your questions.
Thank you so much for your talk. We will be now opening a short Q&A session. So if you have any, well, we hope you have some questions. So if you have any questions, please raise your hand, and Janek will come to you.
Can you hear me? Yeah, so thanks for the talk. I wanted to ask a question about this early stage platynerase larva. When you have this direct connection between the sensory cell and the ciliated cell, it looks like a very basic instance of a reflex arc, but is it really like in automatic reflex arc, or there is some modulation that, for example, depending on some other factors or internal physiology, those connections will work differently?
Yeah, this is a very, very good question. So we don't believe it's just a very simple reflex arc. So just this photoreceptor expresses, for example, neuropeptides, neuromodulators, and also for a reflex arc, basically there's probably no reflex arc that is not modulated in nature. So even the knee-jerk reflex is highly modulated, and the output, the strength of the output is very context dependent, and we believe it's also true here. There is no neuronal, other neuronal connections which could be modulators, but we think that there is modulation which could be, for example, allow the system to tune for intensity or contrast in the visual field. So it's, but yeah, it's as close as you can get to a reflex arc, but reflex is a very misleading concept because it implies this direct sensory motor response, right? That the nervous system's function is just to respond to outside stimuli. And even in this simple case, actually, you really need the movement of the system. So you need the helical swimming, which is controlled by other neurons. And then, so you need the whole body, and you need this embedded basically cognition for the system to work. So just by a reflex, it will never work at all. So you need the movement, and then the ongoing movement is tuned or modulated by the light input. That's a better view of looking at the nervous system, that there's ongoing activity and the sensory inputs modify this activity, then think in terms of reflexes. And we could also show it for the simplest of all circuits with one neuron.
Thanks.
Excellent, really interesting talk. What is the transmitter involved in that simple circuit between the sensory and the cilia?
Yeah, this is acetylcholine, so it's a cholinergic photoreceptor, and we could show if we block cholinergic transmission with bungarotoxin and other nicotinic antagonists, then we can completely block this response. So we believe these are cholinergic synapses, and there are cholinergic nicotinic receptors in the ciliated cells, and those then depolarize the ciliated cells, that leads to calcium influx that we know can tune the stroke of the cilia.
Yes, fascinating. Do you think that calcium might be doing any kind of second messenger work?
Yes, so calcium has a strong influence on ciliary axoneme beating, and depending on the rate of change and the level of calcium, you get all kinds of effects of the cilia. If calcium goes up very quickly, then the cilia stop beating, and if you just have a slower rate of increase, then the cilia start to beat in a different stroke, which reduces the flow of the cilia on the illuminated side, and this is how you get the turns. So there, calcium is a very important second messenger and modulator of cilia.
Fascinating work. Fascinating work.
Thank you.
Okay. Do we have any more questions?
Yeah, thank you for the talk. My question could be totally off. It will be actually not a question. But like, are you familiar with Michael Levin's work and his work on eye creation in planaria, and how these cells recruit other cells to create a functioning eye?
Like in regeneration of planaria and head.
Yeah. And
Yeah.
actually, I transplant to put the eyes into different part of the body. And if there is not enough eye cells, then other cells become eye cells.
That's fascinating. Yeah. We I know Mike Levin and some of his work. I wasn't aware of this transplantation experiment. I should look this up. That's very interesting. We cannot do similar ones. If you transplant here, you basically blow up the larva. But that's that the eye has this kind of developmental signaling role, some kind of instructive role. That's very interesting to make differentiation of the downstream circuit. So I have no idea how that works. We are not working on planaria, but other flatworms, and at least we will try to get the visual connectivity graphs so that at least then you can have the cell types and the connectivity map, and maybe look into these types of situations at a more circuit-aware approach, because it's not known in planarians what are the interneurons and what is the neuronal underpinnings of vision.
Thank you for your talk. My question may also be a bit off topic, but as far as I know, complex camera eyes evolved many times in animals. And judging, for example, by the type of light-sensitive molecules, did these proto-eyes also evolve many times independently, for example, in annelids and vertebrates? Or is it possible that the common animal ancestors also had this kind of structures?
That's a very exciting question, very interesting. And yes, for camera eyes, we know they evolved complex camera eyes at least 3 times. So the vertebrate eye, the cephalopod eye, and some of these annelids, these pelagic annelids, the Alciopid annelids have complex camera eyes. And there we know these were 3 independent, very clearly independent evolutions. And what also helps to trace the history of these structures is, of course, the phylogeny, but also the type of photoreceptors. So the vertebrate eye is ciliary type photoreceptors. Our eyes, the rods and cones, the cephalopods, they have rhabdomeric eyes, and also the annelids have rhabdomeric eyes. And if we look at the simple eye spots across larvae, there are also different types. They are rhabdomeric types, they are ciliary types, and there is a third type which is called the xenopsin expressing type. But it seems to be, from reconstructions, we believe at least now that there were 3 types of photoreceptors ancestrally present, at least in the bilaterian last common ancestor, ciliary, rhabdomeric, and potentially xenopsin expressing one. And also we can trace the history of some of the pigment cells. So probably 1 or 2 type of pigment cells was also there. And then which pigment associated with which photoreceptor is still an open question. But through looking at cell type evolution and also comparative circuit analysis, such as I showed for between flatworm and annelid, we believe that eventually we will be able to reconstruct to the cell type and maybe circuit and maybe functional level what was there initially, and whether these are all independent events. Probably not, because the opsins are all conserved, and they are these 3 major types of photoreceptors with their characteristic opsins that go back even before the bilaterian origins. So it's not completely all independent.
Okay, do we have any more questions? Maybe a last question? Okay.
Hi, thank you for the talk as well. It might be off topic again, but where would you put the eyes of hunting spiders on the graph of the evolution of eyes? Because I know that they're quite complicated and interesting. For those who don't know, some hunting spiders have 4 or even 5 colors that they see in, and they also see most of the. Oh, sorry. They see most of the everything they see is black and white, but they have a little scope in the middle that is in those 5 or 4 colors, and It's just the crazy animals, really. Do you know anything about them, and can they even be placed on that graph, or?
Yeah, the fascinating animals, and I think they have. They may have the characteristic compound eye, but these hunting eyes are different. I think it's some kind of lens eye. So definitely they would go somewhere into the mid-resolution visual systems, and so they can be placed in the graph. This is just a very conceptual graph of resolving power, and they need a vision to hunt, maybe not very high resolution. I could imagine for hunting you need very good motion detectors. That's maybe more important than very high resolution. You need very high resolution for flying hunters, like the dragonflies, which have, of course, the highest resolving eyes within the insects. But yeah, I don't know very much about the details of the eyes. There are so many then elaborations. We have eyes which are mirror eyes with a parabolic mirror. Some mollusks have them. Then within the spiders, large diversity, and so on and so forth. So thank you for the question.
Okay, so I think we will close now our Q&A session. Let's thank again Professor Jékely for joining us today, despite the issues. And we will meet after a short break, after 7 minutes, I think, at 10:50. And we will listen to the next talk. You can enjoy some cookies and tea and coffee outside. And yeah, let's meet in 7 minutes. Thank you very much.
Thank you very much. Bye-bye. Thank you.